Hi everyone, welcome back to Chapter 12. This is our third and final lecture for Chapter 12. We'll wrap up nervous tissue today, and next class we will begin talking about muscle tissue. So let's start with a not really attendance question today, and this one's going to be a little bit tougher. This is more of a critical thinking question, so pause the video and try to figure this out on your own before we talk about what the answer actually is. When an action potential is passed to a neuron, it always moves from the end with the soma towards the end with the axon terminal. It never stops, turns around, and goes back towards the soma. Why? So pause the video and try to answer that, and then we'll talk about what is actually going on here. So here is a neuron, and what we're saying is the action potential comes in at this end where the soma and dendrites are, and it travels down the axon toward the axon terminals, and it leaves. It doesn't ever get halfway down the axon and then decide to go back the other way. That never happens. So, why not? Well, remember when we talked about all of the events happening during an action potential? It's happening in a very small section of the axon, and then that spreads to the next section of the axon, and then that spreads to the next section of the axon, and it just keeps going like dominoes. But when it happens in one section of the axon, what all is actually going on in that area? Remember what channels are opening and closing, and add that to when we talked about why an action potential can't occur in a certain area. Remember we said that if the voltage-gated sodium channel was still closed, we were in something called the absolute refractory period. Well, when we go from one section to the next, when that action potential spreads to the next area, well, this area is now in an absolute refractory period. So it can't receive an action potential. It's kind of like a roadblock that prevents the action potential from turning back and going the other way. It forces the action potential to follow a one-way path down the axon. Okay, if that doesn't make sense, think it through for a little bit. If it still doesn't make sense, send me an email or a text or talk to your tutor and make sure to figure out exactly what's going on there. Okay, so what we're going to be talking about today are how neurons communicate with other neurons or other cells in general. And if we think back to everything that we talked about in the last lecture, when we looked at action potentials traveled down the axon, and it was a continuous path. There was nothing open. It just simply traveled down the path like you're driving down a highway. But when you get to the axon terminals, and then there's a gap before we get to the next axon or muscle cell or gland or whatever cell we're communicating with, we run into a problem. Imagine you're driving down the highway and there's a drawbridge that's open. Well, now there's a big open space between your side of the road and the next side of the road. And that's what synapses kind of resemble. It's a space, a gap between one neuron and the next neuron or the next cell in line. So we're going to see how that action potential can actually still pass even though there's a gap in the road. But before we talk about that, let's talk about a couple of different types of synapses. And a synapse is just where a neuron is communicating with another cell. Most of what we're going to be talking about today are chemical synapses, and that's what I was just describing to you, where there is a gap in the road. But there's also something called an electrical synapse, which is what we see here in this image. An electrical synapse is where a neuron, or in some cases a muscle cell, is connected to another neuron or another muscle cell using gap junctions. Remember gap junctions from back in chapter 3 when we were looking at the plasma membrane. It might have actually been the tissue chapter. But gap junctions, remember, are just tunnels. They're passageways that pass from one cell into another cell. So everything that we were talking about last class, we were talking about ions and movement of ions. And that movement of ions is what electricity actually is. So in action potential, ions in this cell could just pass through these gap junctions into the next cell. That is an electrical synapse. Some neurons do this. Next semester in Bio 139, when we're talking about the heart actually uses electrical synapses. But most of what we're going to be talking about today are chemical synapses, and there's a little bit more to the process there. When we're talking about chemical synapses, we're going to be talking about those ligand-gated channels. And here's a picture that we looked at last class. Remember, ligand-gated channels first are closed and nothing can get through. But when a ligand binds to one of these ligand-gated channels or chemically gated channels, it opens that channel and then ions can pass through. And there's different ligands, and those different ligands open different channels, and the different channels let different ions pass through. For example, some ligands may open a channel that lets sodium pass through. Others may open a channel that lets potassium pass through. And we're going to have different effects depending on which ligand is used, which channel is open, and which ions move through that channel. Now, while we're talking about synapses, there's actually a few different ways that a synapse can be structured. It's always going to involve axon terminals. But on the receiving cell, the cell that this neuron is communicating with, those axon terminals could actually connect at different points. For example, right up here in the top right, we see something called an axodendritic synapse. And this is the one that most textbooks talk about when they're doing the general synapse. But an axodendritic synapse is a synapse between the axon terminals of the sending cell and the dendrites of the receiving cell. Very, very similar, there's something called an axosomatic synapse. And this is a synapse between the axon terminals of the sending cell and the soma of the receiving cell. So here we can see they're just connecting directly to the cell body, directly to the soma. And finally, there's this weird synapse between the axon terminals of the sending cell and the axon of the receiving cell. So this is a little bit different because remember, the axon is the sending portion of a neuron, not the receiving portion, although in this case it's acting as a receiving portion. Here in this image, we can see that it's actually at the axon hillock, but it could be at any point along the axon, even at the axon terminals of this receiving axon. Now, which type of synapse would be most likely to result in an action potential in the receiving neuron? Now, this is actually going to be talked about throughout the lecture today, so we're going to go over what the answer is here. But at the end of the lecture, you'll actually have a little bit more information that might help this make a little bit more sense. Well, up here in the axodendritic, over here in the axosomatic, both of these areas develop what we called graded potentials or local potentials. And remember, graded potentials will affect a bigger area depending on the amplitude or the magnitude of the stimulus. So, if we are sending more and more and more action potentials, then we're going to have a bigger and bigger and bigger effect. Same thing over here, bigger and bigger and bigger effect. But we don't get an action potential here. We just get graded potentials. We'll see why throughout the lecture today. Over here in the axo-axonal or the axo-axonic, different books will call it different things. Here, we only have to get to threshold. Remember, negative 55 was threshold, and at that point we got an action potential. There's something special that happens right here. Remember we said something about the axon terminal? About the axon hillock. In the axon hillock, we only have to get to that negative 55 threshold because the axon hillock is who decides if we get an action potential. The axon hillock decides if we get an action potential. We will see how he makes that decision a little later today. But to answer the thinking cap question, the axo-axonal or the axo-axonic is the most likely to produce an action potential because we only have to get to threshold. Up here, we have to get much higher than threshold so that the area of effect reaches the axon hillock. So we'll come back to this and discuss the whys if that still is a little fuzzy. I understand that it probably is a little bit fuzzy right now because we haven't talked about the axon hillock in the level of detail that you need to really be able to answer this question, but by the end of the lecture today, it will make a lot more sense. But we're now going to switch gears. We're going to draw exactly what's happening at a chemical synapse to see just what happens to pass that action potential from the sending neuron to the receiving neuron. So, what we've drawn here is kind of what is seen in figure 12.23 in your textbook. It's drawn a little bit differently, but it's going to show us the same stuff. So while you're drawing this out, I'm going to explain everything that we're seeing here. This whole picture is a synapse. Now, over on the left, this is the axon terminal of the sending neuron. And everything that we talked about in the last lecture with the action potential traveling down the axon is happening all along this membrane here of this axon. This swollen area here is the terminal button or the terminal bouton of the axon terminal. Inside this terminal button, we see lots of vesicles containing neurotransmitters. Neurotransmitters, remember, are how neurons communicate with other neurons. In this example, we're using the neurotransmitter acetylcholine, ACH. Now, this could be lots of different neurotransmitters, and they're all going to work the same general way, but they will each have different effects. Acetylcholine is the one that textbooks always use in their examples. Acetylcholine is the most common in just general aspects of the nervous system, and it's also what is used in the somatic nervous system. Now, we have all of those same proteins that we talked about before, the voltage-gated, the leaky, the sodium-potassium pump, all of those here along, but we're not drawing that out. We've already seen how those work. When we get to the terminal button, there is a new type of channel that we haven't talked about yet, and it is a voltage-gated calcium channel. Voltage-gated calcium channel. Voltage-gated calcium channels open at threshold, so at -55. So when the action potential arrives at the terminal button, then this voltage-gated calcium channel is going to open. All right, over on the right side of the screen, this is the receiving neuron. This is the neuron that we are communicating with. And what I've drawn here, this is a dendrite, but it doesn't matter if it's a dendrite. It could be a soma. It could be an axon. It doesn't matter. But in this example, it's the dendrite. And we have here one of our ligand-gated channels, or a chemically gated channel. In this case, it's a ligand-gated sodium channel. Now, the whole thing, like I said, is a synapse. The open space between the two cells is called the synaptic cleft. The sending neuron that comes before the synapse is called the presynaptic neuron. That's the sending neuron. The receiving neuron, it comes after the synapse, so it's the postsynaptic neuron, the receiving neuron. We still have the same concentrations of everything that we've talked about. Sodium is high outside the cell, low inside the cell. Potassium is high inside the cell, low outside the cell. Calcium is high outside the cell and pretty much non-existent inside the cell. Remember, calcium, if it's inside of a cell, it's stored in the smooth endoplasmic reticulum because calcium likes to attach to things. And that's generally not something that we want. So we keep calcium stored away outside the cell or in the smooth ER inside the cell. So now let's put things in action and see exactly how we get an action potential from this presynaptic neuron to the postsynaptic neuron across this open space, the synaptic cleft. Well, the action potential that we were drawing yesterday is coming along down the axon, and eventually it gets to this terminal button. And once that action potential gets there, as long as we reach threshold, which we will, then the voltage-gated calcium channel is going to open. It opens at threshold. So now there's an open pathway for calcium to pass through. Which way is calcium going to go? Well, everything likes to go from high to low concentration, so calcium is now going to rush into the cell. We've got a lot of calcium inside the cell now. But what did we just say about calcium? For the most part, we don't want calcium inside the cell. Why? Because it likes to attach to things. And that's what it's going to do. Calcium attaches to these vesicles. Calcium attaches to the vesicles that contain neurotransmitters. And when calcium attaches to a vesicle, it causes them to migrate towards the cell membrane. Think back to when we were studying the cell. When a vesicle containing something bumps up against the cell membrane, what does it do? We get exocytosis. So all of that neurotransmitter, in this case acetylcholine, is exocytosed. It's dumped out of the cell into the synaptic cleft. So now there's a really high concentration of acetylcholine here right up against this terminal button. And there's practically no acetylcholine throughout the synaptic cleft. What's going to happen? We get diffusion. The acetylcholine spreads across the synaptic cleft. Well, now we haven't used the word in this instance yet, but acetylcholine, it's a ligand. And it is the ligand for this ligand-gated channel. So acetylcholine diffuses across the cleft and attaches to these ligand-gated sodium channels. What happens when a ligand attaches to its channel? It's like a lock and a key. It causes the ligand-gated channel to open. This is a ligand-gated sodium channel. It opened. What happens to sodium when a channel is open? It's going to pass through down its electrochemical gradient from outside the cell to inside the cell. And when sodium is coming into a cell, what happens to that cell? It's going to depolarize. And we start causing a graded potential here in the postsynaptic neuron. We say, exciting the postsynaptic neuron. We're taking it closer to threshold. All right, now before we get off of this picture, I want to talk about some variations. In this case, we used acetylcholine, which opened a ligand-gated sodium channel. But different neurotransmitters will open different ligand-gated channels. What if instead of a sodium channel, what if it was a potassium channel? Potassium would go out of the cell, taking his positive charges with him, making the cell more negative, or hyperpolarizing the cell, taking it further away from threshold. That makes it less likely that this cell will send an action potential. So different ligands, different neurotransmitters, will open different ligand-gated channels, which let different ions pass through, which then have different effects on the postsynaptic neuron. So here is your book's way of showing exactly what we just drew. Here is the axon terminal. We're coming down. We get to the terminal button or the terminal button. We've got these vesicles containing neurotransmitter, probably acetylcholine. When that action potential comes down, voltage-gated calcium channels open. Calcium comes in, attaches to these vesicles, causes them to travel to the edge of the axon terminal button, exocytosing the neurotransmitter, which then attaches to these ligand-gated channels. In our example, sodium came in, depolarizing the postsynaptic neuron. Here is a different textbook's way of showing it, and it actually shows all of the steps that we just talked about. Binding of neurotransmitters opens ion channels. So Maybe we get sodium coming in. Maybe we get potassium going out. Maybe it's a completely different ion coming in or going out. All we need to think about is what is it doing to the receiving cell? Is it causing it to become more negative or less negative? Is it going to cause it to depolarize or hyperpolarize? So I don't think this is going to have sound, but this is on the PowerPoint that's on Blackboard. So I'm going to play this video and we're going to see everything that we just talked about in action. Okay, so that was already posted on our PowerPoint. So if that didn't have sound, which I don't believe it ever does, still watch that video through the PowerPoint where it will have sound. It just kind of animates everything that we just drew. Now, like I said, there's lots of different neurotransmitters. You do not need to know them all. You don't need to know their effects. I would like you to know the effects of the two that I have listed here. Acetylcholine, which is what we used in our animation or our example, it opens voltage-gated sodium channels, and we saw that that depolarizes the receiving cell, the postsynaptic neuron. And because of that, it makes it more likely to send an action potential. There's something else called GABA. GABA opens voltage-gated chloride channels. And remember, chloride is Cl-, and we saw that it was higher concentration outside the cell. So if GABA opens, I'm sorry, I said voltage-gated, I meant ligand-gated. GABA opens ligand-gated chloride channels, so chloride goes into the cell. And if chloride, with his negative charge, goes into the cell, what's going to happen? It's going to hyperpolarize that receiving cell. It's going to make it less likely to generate an action potential. So we can say in ACH's case, ACH is excitatory, and GABA is inhibitory. Excitatory makes the receiving cell more likely to send an action potential. Inhibitory makes the receiving cell less likely to send an action potential. So there we can see how different neurotransmitters will have different effects. But once that neurotransmitter has an effect, we need that effect to end. We don't want that effect to just keep going. So how do we make sure that that signal stops when we need it to? There's a few different ways. There's something called reuptake, and that's just the sending cell pumps the neurotransmitter back into itself. It takes it back up, it reuptakes it. Now, sometimes this happens a little bit too quickly. And if it happens too quickly, that's basically saying as soon as the neurotransmitter is released, it's pumped right back in. And we don't want that. We need the neurotransmitter to stay out in the synapse long enough to have an effect. So sometimes this is actually going to become a problem. For example, some types of depression, the neurotransmitter serotonin is released and then is uptaken too quickly. There is actually a class of drug called a selective serotonin reuptake inhibitor, or SSRI. Selective serotonin reuptake inhibitors selectively inhibit the reuptake of serotonin. It blocks the reuptake a little bit so that the serotonin stays out in the synapse long enough to have its effect. Degradation. There are enzymes out in the synaptic cleft that break the neurotransmitter apart. And if it's broken apart, then it doesn't have the ability to cause an effect anymore. So after the neurotransmitter travels across, after it has its effect, the enzymes will break it down. And there's different enzymes for different neurotransmitters. In the case of acetylcholine, there's something called acetylcholine esterase. Acetylcholine esterase takes acetylcholine, breaks it in half into acetate and choline. And the choline is then what is reuptaken, in this case, so that the sending cell can make more acetylcholine from it. Simple diffusion. Well, when the sending cell stops sending acetylcholine, or whichever neurotransmitter, we stop having that concentration gradient. So the acetylcholine stops diffusing across, and instead we start having the reuptake cause the gradient to go in the other direction. So now we diffuse away from those ligand-gated channels. And it's not that one of these things happens, it's all of these things happen. When we release this neurotransmitter, we start to get degradation by the enzymes, reuptake of the components, which allows for diffusion away from the ligand-gated channels. And that will then end the signal. So we kept saying that different neurotransmitters will have different effects. And sometimes they're excitatory. Sometimes they bring the receiving neuron closer to threshold and make it more likely to send an action potential. Sometimes they hyperpolarize the receiving neuron, taking it further away from threshold and less likely to send an action potential. So we're going to give names to those. If they are excitatory, we say that we get an excitatory postsynaptic potential developing. If it's inhibitory, if we hyperpolarize, then we get an inhibitory postsynaptic potential developing. So EPSP, we're exciting the postsynaptic neuron. IPSP, we are inhibiting the postsynaptic neuron. EPSP, more likely to send an action potential. IPSP, less likely to send an action potential. In our acetylcholine example, that was an EPSP. In GABA, that's an IPSP. Different neurotransmitters will have different effects. And in some cases, different neurotransmitters will have different effects in different locations in the body also. But typically, we don't get a single action potential from one neuron to the next. We actually have lots of action potentials coming to the synapses, and then those synapses sending multiple signals to the receiving neuron. And when that happens, the effects are going to stack. We say we get summation. And summation comes in two different forms. Temporal summation is when one presynaptic neuron is sending multiple signals to the receiving neuron, one right after another, after another, after another, very quickly in time. Another way is spatial summation. Spatial summation, multiple neurons are acting as presynaptic neurons, and they're all sending signals at the same time to a postsynaptic neuron. In both cases, the signals stack. They add together. They have summation. We kind of saw that at the end of that video that we just watched. But here is an example of of temporal summation. One neuron sending a signal and then another signal and another signal very quickly, one right after the other. This is temporal summation. Each of these black arrows point to when the action potential arrives at the synapse and is then sending those neurotransmitters to the receiving neuron. Here we have one, and then before we come back to rest, we get another. And before we come back to rest, we get another. And they keep stacking, each time bringing it closer and closer to threshold. Once we reach threshold, that's all that it takes. We get an action potential developing in the receiving neuron. Now, let's go back to this. Even though it doesn't show it, we could have spatial summation, some EPSPs and some IPSPs, and they basically fight it out. Are we going to get closer to threshold? Are we going to get further away from threshold? Do we get to threshold or not? And that's what we saw at the end of that last video that we watched. And sometimes neurons can actually help each other out to make an action potential more or less likely to be passed from one neuron to the next. We call that facilitation. And the facilitation occurs at the synapse with the presynaptic neuron. So what we see here, this is from our textbook on figures 1228 and 1229. Here is the presynaptic neuron. Here is the postsynaptic neuron. And here's what we see, the facilitating neuron. So the facilitating neuron is not sending any information to the postsynaptic neuron. In this case, presynaptic facilitation means that the facilitating neuron has an axonal synapse with the presynaptic neuron. And the facilitating neuron is going to send some neurotransmitters to the presynaptic neuron that causes, in this case, voltage-gated calcium channels to remain open longer. The facilitating neuron sends action potentials to the presynaptic neuron, causing voltage-gated calcium channels to remain open longer. Well, the longer the calcium channels are open, the more neurotransmitters are going to be sent into the synaptic cleft, having an effect on the postsynaptic neuron. So the facilitator helped the presynaptic neuron cause an effect on the postsynaptic neuron. We could also get presynaptic fission. Very, very similar. We still have this neuron communicating with the presynaptic neuron, but in this case, it's an inhibitor. The inhibitor releases some neurotransmitter that causes the voltage-gated calcium channel to remain closed. And if the voltage-gated calcium channel remains closed, then we don't even get the release of neurotransmitters. So we do not have an effect on the postsynaptic neuron. So we are either facilitating or inhibiting the presynaptic neuron so that it has more or less of an effect on the postsynaptic neuron. But the difference here is it is this accessory neuron that's having an effect. It's not the pre, it's not the post. It is an accessory neuron, either a facilitator or an inhibitor. So now let's go back to when we were talking about the three different types of synapses. We said that the axonal was more likely to cause an action potential in the receiving neuron. Let's look at exactly why that is. Now, up here, if we send an action potential to a dendrite, if we send an action potential to a soma, it does not matter how strong of an effect we have. Remember, we get graded potentials. And like we drew in our previous lecture, graded potentials can be very small. They can be a little bit bigger. They can be a little bit bigger. They could be really big. They could be ginormous. The bigger they are, the bigger the area of effect. So something that goes to -20 will have a certain size, but if it goes way up to above +50, like we have in our picture there, it will have a bigger area of effect. But graded potentials, no matter how big they are only local. They are only graded potentials. Another way that I'd like to say that is, if we looked at a dendrite, if we looked at a soma, we will never see an action potential. We will only see graded potentials. Soma and dendrite, no action potentials ever. Even if we reached threshold at the soma or at a dendrite, it does not cause an action potential. But at the axon hillock or any point along the axon, we do get action potentials as long as we reach threshold. And it's all going to come down to this guy right here, the axon hillock. Remember on the first lecture of this chapter, we said axon hillock is who decides if an action potential is going to develop in an axon. How? This is going to take some critical thinking. We never get action potentials in the soma or in the dendrites. The axon hillock determines if we get an action potential. And from the axon hillock onward, we can and do get action potentials. What's the difference? Well, what did we say in lecture 2 of this chapter was the very first thing that happens in an action potential? Who was the key player? Voltage gated sodium channels. Voltage gated sodium channels happen at threshold. They open at threshold. And that sets an action potential in motion, and we get a full action potential as long as the voltage gated sodium channel opens. Well, guess what is not found in the plasma membrane of a dendrite or the plasma membrane of the soma? Voltage gated sodium channels. Guess what is found in the plasma membrane of the axon hillock and all along the axon? Voltage gated sodium channels. Let's tie it all together. If we get a small graded potential somewhere in the axon, it affects a very small area. If we get a bigger graded potential somewhere in the dendrite, not axon, if we get a small graded channel somewhere here in the dendrite, small area of effect. If we get a bigger graded potential here in the dendrite, bigger area of effect. If we get a bigger graded potential here in the dendrites, bigger area of effect. If we get a really big graded potential here in the dendrite, that area of effect is going to be so large that it reaches the axon hillock. We get an action potential develop, and the action potential travels the entire length of the axon until it gets to the axon terminals, and now we're at a synapse. Did that make sense? Graded potentials are only found at the soma and the dendrites. We never get any action potentials at the soma or at the dendrites. The bigger the graded potential, the bigger the area of effect. If it's a big enough area of effect that reaches the axon hillock, then we open those voltage-gated sodium channels and action potential travels along the axon. Now, the types of circuits, that was self-study, but I do want to bring in a thinking cap question related to those types of circuits. If you've ever walked into a place and smelled something from your childhood, you typically have instant, vivid memories. If you see photos of that same time, you'll also have those same memories. Imagine if you walked into your grandparents' house and you smell that smell and you think back to a time when you were a child visiting your grandparents' house. Those memories are instant. If You look at pictures of when you were a kid at your grandparents' house, you can have those same memories. So how can seeing something and smelling something have such similar responses? Which of those circuits is involved there? That's a converging circuit. That's when, for example, images coming from your eyes reach a part of your brain where a memory is stored. Images coming from your nose also reach that same part of your brain where that memory is stored. That's converging circuits. And finally, I want to talk about déjà vu, which is also going to tie into those types of circuits. When you look at the different types of circuits, there was one called parallel after discharge, and it's in your book on figure 12.32. I'll kind of try to draw it out here. We have a neuron that synapses with 3 different neurons. And then those neurons synapse with other neurons. And the action potential from this original neuron is going to arrive at this neuron at the end more than once. Even though one action potential was sent, more than one action potential arrives at the receiving neuron at the very end. But not only does more than one action potential arrive, one arrives very quickly, one arrives a little slower, one arrives a little slower. So we get 3 action potentials, one after another after another. And this can be explained, or it can help explain if you've ever looked at a light and then closed your eyes and you still see the light for a little while. That's because those action potentials from cells in your retina, the back of your eye, are arriving one after another, instead of all at once. But they also suspect that this might be what causes déjà vu. You experience something, it creates a short memory, but then that same exact thing arrives a moment later. And even though it's just a moment later, they think that there might be something where your memory that had already developed makes it seem like that memory was from a lot longer ago, even though it just happened. So, these parallel after discharge circuits, they think can explain déjà vu. And that's just because one action potential arrives multiple times at the receiving neuron at the very, very end. Okay, so that wraps up nervous tissue. We're not done with the nervous system, actually. It just wraps up nervous tissue. Next, we're going to start talking about muscle tissue. And muscle tissue actually uses a lot of the same concepts that we learned in this chapter, so make sure they make sense. Ask questions if they don't. After we talk about muscle tissue, we're going to come back and start talking about the nervous system. So things like the brain. Okay? So make sure this all makes sense. Ask questions. See me, see your tutor, whatever it takes. But make sure to get any questions cleared up. All right? Take care, and I'll talk to you next time.